This is the spliced rRNA intron in tetrahymena. This spliced rRNA intron in tetrahymena has 414 nucleotides. This boxed sequence is called as internal guide sequence. The internal guide sequence has the cleavage site. First of all, removal of 19 nucleotides from 5' end of spliced rRNA intron in tetrahymena occurs. As a result, L19 IVS is produced. L19 IVS is a ribozyme. This L19 IVS has 395 nucleotides. A portion of internal guide sequence remains at the 5' end of L19 IVS. The G residue added in the first step of splicing reaction is part of the removed sequence. L19 IVS lengthens some RNA oligonucleotides at the expense of others in a cycle of trans-esterification reactions. The 3' OH of the G residue at the 3' end of L19 IVS plays a key role in this cycle. This is not the G residue added in the splicing reaction. A synthetic C5 oligomer is one of the ribozyme's better substrates. Because the synthetic C5 oligomer can base pair with the guanylate-rich internal guide sequence remaining in the intron. During the cycle of trans-esterification reactions, first of all, a C5 oligomer base pairs with the guanylate-rich internal guide sequence. The hydroxyl group attached to 3' guanine attacks on the bond between two adjacent cytosines in the C5 oligomer. Due to this attack, the hydroxyl group attached to 3' guanine makes bond with the cytosine of C5 oligomer, and the cytosine attached to a hydroxyl group in C5 oligomer makes bond with the 3' guanine of L19 IVS. As a result, a C4 oligomer is produced. The C4 oligomer is then released from the L19 IVS. A C5 oligomer then base pairs with the guanylate-rich internal guide sequence. The hydroxyl group attached to cytosine of C5 oligomer then attacks the bond present between 3' guanine and its adjacent cytosine. Due to this attack, the hydroxyl group of C5 oligomer makes a bond with 3' guanine of L19 IVS, and the cytosine with its attached hydroxyl group makes a bond with the cytosine of C5 oligomer. As a result, a C6 oligomer is formed. This C6 oligomer is then released from the L19 IVS. Due to the release of C6 oligomer, L19 IVS becomes free to bind with another substrate. Now, some important points. Each ribozyme molecule can process about 100 substrate molecules per hour. L19 IVS follows Michaelis-Menten kinetics. L19 IVS is specific for RNA oligonucleotide substrates. L19 IVS can be competitively inhibited. The Kcat by Km specificity constant of L19 IVS is 10 to the power 3 mole per second. L19 IVS accelerates hydrolysis by a factor of 10 to the power 10 relative to the uncatalyzed reaction. L19 IVS uses substrate orientation, covalent catalysis, metal ion catalysis strategies to convert the substrates into products. The catalytic activity of L19 IVS is irrelevant to the cell. The catalytic activity of L19 IVS has important implications for current hypotheses on evolution. Please like, subscribe and share.